Hello and welcome to the Neuron Regeneration Podcast. My name is Jared Carter. Thank you so much for joining me. Today's episode looks at a potential therapeutic approach to reversing Parkinson's disease. Now that sounds like a really bold statement, but to date, all of the treatments for Parkinson's are predominantly aimed at symptoms and reducing symptoms. And there have been some absolutely amazing breakthroughs. My own grandfather was part of the early clinical trials for deep brain stimulation, and he had an incredible outcome. It added massive quality to his life, multiple years to his life. But whether therapeutic or device-based, none of the treatments to date have been able to stop the progression of the disease, let alone reverse it. And as you'll hear from our guest in this episode, he has a very, very different approach to treating Parkinson's that has actually shown in multiple cellular and animal models to reverse the pathology, and has been given the thumbs up by the FDA to enter human clinical trials. Now, he gives his background in the beginning of our conversation, but I do want to read just a little snippet from his bio. Jonathan Sackner-Bernstein, MD, studied engineering prior to his career as a physician. He served on the faculty of Columbia University while working in clinical care and clinical trials focused on heart failure. His expertise in drug development is based on expertise built in industry, at the FDA, and at DARPA. And Sackner-Bernstein's mission of conquering Parkinson's started when his friend and mentor began to suffer from worsening Parkinson's disease. And I'd just like to add that with his really diverse background, he brings a very broad and non-siloed approach that I think brought out some great insights that we can apply to neuron regeneration research as a whole. And I think anyone interested in this topic really should listen for those. We kind of dig into those towards the end of our conversation. And finally, if his work or Parkinson's disease in general is a specific interest of yours, definitely check out his blog at parkinsonsdisease.blog. And now, please enjoy my conversation with Dr. Jonathan Sackner-Bernstein. Jonathan, such a pleasure. I'm so excited to have this conversation with you. Thank you for joining.
Well, thank you for having me.
Awesome. So why don't you go ahead and give us a background on your life and your story through medicine and what brought you to your interest and your research in Parkinson's disease?
Well, it's sort of a combination of a professional as well as a personal. So in terms of professional, I went through medical school after studying engineering, learned how to be a cardiologist and doing clinical trials primarily in the Columbia system here in New York, really with the idea of learning how to develop drugs and devices to treat cardiovascular diseases. And that ended up leading me to a path that was a little bit less focused on cardiology and more on how to develop drugs, how to develop devices, how to tell whether they're really impacting people the way you want to, in terms of effectiveness and safety across all stages of development. I spent some time in a senior position at the FDA. I worked in the inner circle at DARPA for several years, and I've worked for a bunch of small to mid-sized companies, either as an employee or as a contractor over the last decade. But this chapter really started in the mid-2010s, around 2015 or so, when a very close friend, Ivan, started to experience the signs and symptoms of Parkinson's disease. And by 2017, he was really starting to go downhill. And he kept asking me to do something to help him. And I really fought against that because I did not feel capable of being able to help figure out how to treat or manage Parkinson's, considering how many people who are really smart have been doing this for decades, despite the fact that there really hasn't been much of an advance since the 1960s, when dopamine-based therapies were first recognized as potentially useful. But Ivan was very pushy, very persistent, and I had a gap in my schedule towards the end of 2017, and I said, fine, just to get him off my back, I'll start reading about Parkinson's disease and I'll show you that I'm useless. And as I started to read about Parkinson's disease, I started to find that there were a bunch of questions where people posed answers. And when I say people, I mean scientists and clinicians posed answers for which I couldn't find data. So, I'm trying to understand what to do and how to think about things. The concept is you go back to first principles. What are the basic assumptions that are the foundation for our current knowledge and our current approach to the disease? So if you look at first principles, there are things like, hey, it's a disease of dopamine deficiency. Now, that's a little superficial, but that's accurate. That's how people talk about the disease. That's how I was taught in medical school. That's how the textbooks talk and write about the disease. Well, we've got, tens if not hundreds of different drugs. Some of them are copycats, some are made slightly differently, but they all are shown to potently and reliably increase dopamine in the brain. So the first question I had was, if this is a disease of dopamine deficiency, and we have untold numbers of drugs and even some devices that can restore brain dopamine levels towards normal, then why aren't these drugs fixing the disease? And they're not fixing the disease. Now, people in Parkinson's, the experts, the clinicians, and the scientists don't like probably when I talk about it this way, but by definition, the treatments that have existed since the 1960s for Parkinson's are palliative. They basically try to help manage the symptoms, but the disease keeps getting worse. So they're not fixing the disease. So that meant to me, well, it can't be as simple as a disease of dopamine deficiency. And that got me started on this course. And I told Ivan, he's like, I told you so. And then I started down this path.
And what did you find as let's just start from the beginning of that path. Is there now data that says yes or no to the dopamine deficiency?
Well, here it depends who you talk to, and how you talk to them. If you were to ask that question to 10 random Parkinson's experts, I would imagine that at least 8 of them would say, no, there's not data. But they wouldn't be acknowledging a whole host of data, which I recently reviewed in a peer-reviewed neurology journal that was published this year, the Journal of Neurology, and which
yeah, for sure. If you could share that with me, we'll make sure we link to it.
Yeah. I will do that. And also, which I talk about in my weekly blog, which is available at parkinsonsdisease.blog, that there is a growing wide range and set of data for which there is not conflicting data. So these data say this is a disease of dopamine excess when you look at the disease from the point of view of what the neurons are experiencing in people with Parkinson's. So traditionally what you do is, what these scientists have done since the 1960s is they would look sort of at, this high level view from the outside. They grab the piece of brain that's relevant to Parkinson's. They'd mash it up into a soup, which means it's combining the fluid that was outside of the cells with the fluid that was inside the cells. And of those cells with fluid, it combines the dopaminergic neurons, which are the key cells that drive the disease or allow you to have normal movement, as well as other cells. And you make a soup, and that soup has a lower amount of dopamine when it's made from Parkinson's people's brains than when it's made from the brains of people without Parkinson's. So these are brains that are donated by people and studied within hours of death. Standard method, nobody really argues with the validity of those measures. So what was learned in the 20 years after that was first discovered, and that was discovered in the 1960s, by the 80s or 90s, we learned that neurons are lost, that neurons become dysfunctional. They lose their interconnecting cables called axons. So if you take into account the amount of axons that are lost, and then also the machinery inside the cell that controls dopamine in terms of whether it's broken down into toxins or used to send signals from one cell to another, if you account for all those things, what you see is the amount of dopamine on average in a dopaminergic neuron inside it, which is what causes function or dysfunction, not what's in the soup of the brain. The amount inside is markedly elevated in people with Parkinson's. So when I first figured that out, I figured that out because I knew we had to understand what the levels were inside the cell, because that determines normal or abnormal function. And nowhere in the literature had anybody ever measured or calculated that number.
And since then, they have been able to.
Well, since then, I have.
Okay.
So I was able to calculate that and publish that in a separate paper in middle of 2021. Okay. And I really only did that, published that, after I already knew there were data showing that reducing dopamine makes sense. So scientifically, it's one thing to show a relationship between an abnormality and disease, or an abnormality and how people feel. And this abnormality of high intracellular dopamine in the neurons, in the brain cells, is interesting, but it's not enough to convince me, and it shouldn't be enough to convince anybody else, that we should reduce dopamine. The way to convince yourself that it's important is to start out by doing laboratory studies and find a way to lower the dopamine levels and see what happens to the disease. Well, it turns out that at this point, there have been 9 different models of disease. Some are animal models, some are cell models, some are models that I find persuasive, some are models that I'm not so convinced by, some are models that people have been using for decades that they believe are really important. It varies, but there are 9 places where the strategy of reducing dopamine has been tested in the lab. And again, I talk about this in my weekly blog from time to time. I try not to be redundant, but on the website, Parkinson's Disease blog, it has all the weekly posts, so you can look through there. That in all 9 models, every single one, using a specific drug that reduces dopamine, reversed pathology. 9 for 9.
It reduces intracellular dopamine levels.
It reduces intracellular dopamine levels, and it reverses disease pathology. So we know in Parkinson's that some of the things that go awry are things like the cells are exposed to oxidative stress. Well, that's reduced. Another is an abnormal protein form is deposited in the cell that prevents the cell from working. That's called alpha-synuclein. So reducing dopamine reduces alpha-synuclein deposition. We also know that the cells die over time. And there are at least 3 mechanisms that have been tested for cell death. Each of those 3 mechanisms, when you reduce dopamine, those mechanisms are blocked and you get improved neuron survival. There's even one model where they looked at what happened to movement. Now, it's not my favorite model, but it's a model used by biologists for decades. It's basically what's referred to as the fruit fly, or the scientific name Drosophila. They put a gene into the Drosophila that causes Parkinson's in people, and it causes basically the fruit flies not to be able to fly. You treat them with this disease and they can fly. So movement is restored. So with even just a subset of these 9, in fact, there are only 3 of them at the time, I went to the FDA and I said, hey, I think this is a reasonable way to treat the disease. Here's the mechanism. Here's the proof of the mechanism. Here's the way we could reduce dopamine levels in people with Parkinson's in a way that protects them. Because if you abruptly cut off dopamine in somebody who's got Parkinson's, they won't be happy. They won't be able to move. It'll be bad. It could be dangerous. So I shared with the FDA how we would do it in a way that we thought made sense from the existing basic science data, and clinical pharmacology data of this drug, which has been used in these 9 models. They were comfortable and they said, okay, submit the application to do a phase 2 trial in people with Parkinson's. So that's what we're working on right now, moving towards starting a phase 2 trial.
But a phase 2, because this is an already FDA approved drug that's shown to be safe in humans.
Exactly. So this is safe in everything except hasn't been tested in somebody with Parkinson's, but that's okay. That's where we want to test it. But we don't have to do any of the phase 1 work. All that can be leveraged. The FDA has ways where you're allowed to leverage that, even if you didn't do it, so that you can skip into a faster development program as a repurposed drug.
I'd love to, if we have time, come back and segue into going through this process for a repurposed drug and what that means with your capabilities of fundraising and getting things moving, as opposed to developing a new drug. But let's talk about a little bit more about like next steps. So where are you in the process with the phase 2? What do we have in the near and far future? I'm sure there's probably some people whose ears have really perked up to have heard that they've seen this, not just a slowing or modifying of the disease, but an actual stop and even reversal of the pathology in these various cell and animal models. It's really fascinating and exciting to hear. So where are you at in the process, and what's the near future look like?
So we were fortunate enough that we're able to close on our first investment, which allowed us to start manufacturing the drug. So that's already started. That should be done within 6 months, maybe less, at which time we can file what's referred to as an IND application. I referred to that a moment ago. IND stands for Investigational New Drug Application. And that's a formal request to the FDA to conduct a clinical trial here in the US. So we're hoping to, if we can stay on schedule, that we would submit that somewhere around the middle of the year. And that would allow us to start enrolling patients in the phase 2A trial by the end of 2025. And from there, we'll know information by the middle of the following year. So really, it's 18 months from now where we're going to know whether all these data in the animal models, which are consistent and positive for this approach, are going to have similar impact in people or not. And if so, it transforms the entire disease.
Totally. Totally. Yeah. I know across the board that translatability from animals into humans it's a big issue, and one where there's not, as high levels of success in general across multiple syndromes. It's just kind of what I hear over time. Just curious, in the world of Parkinson's disease, do those models that you're discussing, have they in the past, like with the things that have been approved for the treatment of the Parkinson's, do they seem to be fairly translatable models? Like in my kids with SMA, we have really wonderfully translatable models compared to a lot of syndromes. Where does Parkinson's kind of stack up when it comes to the models they use and how things have translated into humans?
So important there to discuss this. I'll start out with the caveat of, if you're talking about how well these models predict what a dopaminergic, or a drug that increases the activity or amount of dopamine, does. So if you want to predict a dopaminergic drug, there is effects in people, great models.
Mhm.
But that doesn't mean anything, because we already know what they do, right? So it's sort of like a tautology. You sort of know what that class of drugs is going to do. That's not so important, because we already know that class of drugs is good for palliation, but not much more. So the part of what I did when I was at the FDA, I was the FDA lead on a project between the FDA, DARPA, and the NIH, where there was recognition that animal models, well, my word would be poorly, I don't think that's not the official word we used at the time, but would poorly predict the effects in people. There's the adage that people like to quote, and I've looked for the source and I can't find it, so I think it's made up. But the adage is that Christopher Reeve said, if I were a mouse, I'd be walking already.
Yeah.
Now, I can't find proof he said that, but I think that it's a valid way to think about animal models. And sometimes people say to me, well, what you're doing is very interesting, but I want to see a large animal model. Why don't you do non-human primate study of your drug?
Mhm.
So I said, well, that's fine if you want to torture non-human primates. I have like a little trouble with that, morally, unless it's going to predict what's going to happen, or unless it's an extreme case. Say you're trying to treat Ebola virus and you can create a non-human primate model, and you can't do it in people. You actually could. You couldn't do it in the US, of course, but you could do it overseas. But where it's not possible to do in people, there are ways where animal models are accepted for FDA approval decisions. This is not one of them. And non-human primates, dog models, none of those models are known to predict what's going to happen in people with Parkinson's. So why would you do it? And one of the things that we did in that joint project between FDA, NIH, and DARPA was focus on some new technology, but also acknowledge that the best you can do is either use an animal model to predict one little aspect of risk you're worried about, or show a potential mechanism of action for the drug, or you do a whole wide range of animal models and cell models. And if it's consistent, then you're more optimistic, but you still don't have proof. So I would describe myself as extremely optimistic that this approach is going to work. But you don't know until you do the clinical trial. That's why it's so important for me to find a few more investors, so we have enough money to conduct the clinical trial. Right now, we've got enough money to get ready for the clinical trial, but not enough to do the trial.
And so roughly, how much is it going to take? Like how many subjects, trial participants are you going to need? And what's the total cost expected to do this phase 2 trial?
So we've had feedback from the FDA through 2 rounds of engagement. So I'm pretty confident we know what we need to do in this first trial to be permitted to go to the bigger next trial. So the first trial is going to be around 24 people enrolled to be tested at different drug levels, dose levels, and assess the tolerability and look for clinical impact. This trial will really show, at minimum, if it's successful, that people can tolerate the drug. So if you believe that the disease is a state of dopamine deficiency, and you treat people who are symptomatic with something that reduces dopamine, you should make them worse.
Mhm.
If we don't make them worse, and I don't think we're going to make them worse, because they actually have dopamine excess,
Mhm.
and we therefore are creating an environment that allows the cells that are alive in the brain, the dopaminergic neurons, allows them to start functioning again. So you get some neuron healing, and therefore better neuron function, that we show that, and we've proven that the current model's wrong. It's not dopamine deficiency. And then we do the next study that proves that we've got the right dose to change the disease course.
Okay.
To do that first study is going to take about $5 million. So we still have a significant amount of money we need to raise. We've got a handful of investors who are looking at us and what's referred to in the due diligence stage. So I'm optimistic it's going to happen in the next couple months, but you don't know till the check clears, as the old statement goes.
Yeah. And I maybe this is a good time to talk about the repurposing play, and how does that affect what the investors are going to look at, what they're going to object with, as somebody who might be in a scenario where I'm trying to do something similar in the future. And like right now, with my own kids, we have some drug repurposing kind of discovery projects, not in my kids, but you know what I mean? We're looking at the potentials there and taking them into animal models and then beyond if we have good initial data. And this is on the regenerative side of things, not the disease modifying side. So when it's an established drug, and I think some of the ones we're about to start experiments with are even in generic format at this point, how does that change the conversation and the ability to raise money for human trials?
The first response I generally get from a professional investor, like a venture fund, when I talk about funding the development of a repurposed drug is not interested.
Mm-hmm.
And they'll say that because it really does not fit into the model that they have figured out works for them. Okay. And it's not that they're not open to new models. It's just that they figured out what works. They then go to investors and say, here's what we're going to do. The investors invest according to that model. And you can't get a fund to then go back to investors and say, hey, remember we told you we had all these great platform ideas for new molecular entities. Well, we want to put $30 million, let's say, just to pick a number to develop, past this first study into a repurposed drug. That's a problem. You can't really expect that to happen. There are a couple of funds I found that focused on repurposed drugs, but they're not necessarily the best funded of the venture funds. Mm-hmm. So, we've got those relationships going, and everybody always says the same thing when they're not interested in investing. We want to talk to you when you have that first set of data, because that costs them nothing, of course. I think the key thing to realize when you're dealing with a repurposed drug is you need to have a way to protect the business, because nobody's going to invest the big bucks that you need to get it to market unless they can get their money back. Mm-hmm. I mean, I know that sounds horribly negative about the way people invest, but I think it's the way people invest. They know, especially certainly professionals. Can you get foundations potentially to do it? Yeah, but generally not all the way to approval. It's just too much money. Right. There are exceptions to that, like Cystic Fibrosis Foundation did that. They did it with an arrangement where they've actually gotten money back to the foundation in a way that's really allowed them to fund a whole bunch of research. So, with the right foundation, maybe that's an option. But what we were able to do was first identified this drug that was first approved in 1979. So it was generic, as you're talking about, which means that anybody can pretty easily develop a manufacturing process, file with the FDA, and start selling a generic. But what we were lucky enough to stumble into is that the dosing form that exists for the approved drug and the generics is so much higher than what a Parkinson's patient is likely to tolerate, that we had an opportunity to propose a novel dosing regimen with smaller doses and different formulations and applied for that patent. So the patent was not just for method of use, which you can get, but then practically speaking in the claims has some information about the novel dosing regimens that we would use based on pharmacology data that already existed. And that's been granted.
The patent is a method of use patent only.
It's a method of use patent, but in the claims also has novel dosing claims. And those novel dosing claims basically block out anybody who wants to file a generic or the holder of the original approval because their dose is too high.
If they were to file a generic, they couldn't just be like, well, I'm going to do the generic, but at a tenth of the size of the dose in pill format or whatever. So you guys kind of, you have the rights for that specific dosage or that range of dosages if this works.
For the range of dosages that are lower than what they have approved. And they can't just go back and file a new application. They'd have to generate clinical data. If they tried to do it in Parkinson's, they'd be violating our patent. Once we're on the market and we have this patent, the FDA regulations stipulate that the FDA could not accept another application. even if company X went out and did the same studies, copied the protocol, did it in Parkinson's, and got the same positive results that led to our approval, the FDA wouldn't accept their application, so they couldn't market it. They couldn't manufacture it in the smaller doses or market it.
Very
So
interesting.
we figured out a way to box them out.
That's
And I
very
think that you need to have something like that, or there's no way to protect the franchise. And then you can prove the concept, but you could run into problems that I've seen with people developing generic drugs for an indication, generating data so that people with a genetic disease say, oh my gosh, this could be great for my kid. I want to use it. And the pharmaceutical companies basically charge enough money to make it worthwhile. For an ultra orphan indication, you'll charge $25,000, $30,000 a month.
Mhm.
And because it's not FDA approved, the insurance companies don't cover it.
Right. Okay.
So it doesn't really do much good for the public health,
Right.
even though there are some people who, thank God for them, can afford to do it. But that's what you run into if you don't have a way to protect your business model.
That's very interesting. If the dosage is significantly different enough that you have a pathway forward in this kind of quote unquote drug repurposing play. And I feel like we're going to have so much opportunity there with AI, being able to connect the dots and see that this obscure, drug for XYZ indication could really be positive for something that people have been waiting with no answers, or limited answers. So I think we're going to see a lot of that in the coming, decade, where this question mark of how to get funding for drug repurposing scenarios, that's cool to hear that this is a pathway that you guys have found forward. So well done on that. Just curious to kind of, I know that what you're doing being somewhat the opposite of what's been the generally accepted approach, right? I'm sure that there's been a lot of pushback. And just to play the devil's advocate, what are the primary objections that you get from the medical or scientific community? And, what are your responses to those primary objections?
Well, the first thing that people will say first of all, I try not to tell people I'm reaching out to initially that I want to reduce dopamine, because I generally don't get answers if I leave that voicemail or send that email. It's just like nothing, no traction at all. So I talk about the novelty. I'll talk about the fact that I have a way to treat disease at the presynaptic neuron as opposed to the postsynaptic neuron. And by that, I mean there's a neuron that has the signal first. It connects to a second neuron, and that's the postsynaptic, because synapse is what's in between the two neurons. So I'll talk about it that way. Once, they hear that I want to reduce dopamine, a very common response is, that's going to hurt people.
Yeah.
And I'll say, well, actually, I think I also figured out a way to do it where it's not going to hurt people. And if I'm lucky, they'll say, well, what's that? And I'll say, well, first of all, let me explain to you that I shared this with the FDA and they felt comfortable. Their only response was, we want to make sure you counted according to that definition that you just proposed. It's like, okay, that's fine. That's why I proposed it. I want to count any episodes that meet that definition that could reflect that I'm hurting people or putting people at risk of being hurt. But it's hard to get people to dig into the data with me. And generally it doesn't happen on the first time or the second, sometimes not the third. There are people I've talked to 10 times who I'm getting close to the point where I think they'll engage with stuff. I'm not trying to convince everybody now. I'm really trying to find people who I think could be allies. So it's, if somebody says no to me 9 times, I generally don't go for the 10th. But if it's somebody who's got some really interesting experience or insights and they're getting warmer, I'll keep trying. But my goal is to get enough people around me so that we can get this first study done. And then with those data, I think it's going to be a lot easier. I think that way because when I was a fellow in cardiology, I was lucky enough to basically be one of the team that started the first phase 2 trial of a beta blocker for heart failure. It was called carvedilol, the brand name Coreg. And this was really crazy at the time. There were people who were telling me, this isn't such a good idea for your career because this is going to hurt people. And sure enough, there's data to show that you can hurt people with a weak heart, which is what heart failure is thought of, certainly at that time of day. We know there are other causes, of course, but that's a big one. If you give a beta blocker at too high a dose. But we learned that if you start at a low dose and slowly increase, people will tolerate it and see benefits within a couple months of starting it. Like meaningful benefits, reversal of disease course, restoration of heart function. I mean, there are a lot of things where starting a drug can be risky, but it's good in the long run. I mean, if I had leukemia, I want chemotherapy to kill all the cells in my bone marrow. It would mostly kill the really fast, quickly dividing cells, which are the cancer, but it would kill the good cells too. And then I'd wait with my fingers crossed that the good cells were the only ones that came back. I'd have that toxic effect. So heart failure is an opposite. Insulin, when it was first developed, was the opposite. The standard of care in the early 1900s was to basically starve kids. If you go back and look at the historical reports of the state of diabetes care starting in, as insulin was about to reach the market, you'll see these kids, these 6-year-old, 8-year-old kids, who look like they've literally been gone through trying to starve them to death. It was the only way to keep them from dying from diabetes. Some of them then had follow-up pictures a year after starting insulin, and they were eating and having insulin, and they look like your normal 8-year-old. I mean, it's really dramatic, but that was a complete shift. Yeah. There are a number of cases where either a shift or getting back to the way, I know you think about trying to get silos broken down between experts is so important by getting, an Australian gastroenterologist and pathologist got together and thought, hey, could it be that gastric ulcers in the stomach, could be caused by a bacterial infection? They were thought as being crazy. Mm-hmm. And, thinking about the word crazy a little differently, actually, I think they were crazy because what they ended up doing was infecting themselves with the bacteria, getting endoscoped, then infecting themselves, getting endoscoped, showing that the bacteria are there, treating with antibiotics, then getting another endoscope and showing that it was cleared. I mean, look, they did win the Nobel Prize for the work, but that's a little extreme. I'm not planning on giving myself Parkinson's.
I love this series of examples where what was thought to be the case with whether it's treatment or pathophysiology or both, that it was actually the opposite to some, fully the opposite or quite different. And, I mean, fingers crossed, this is another one of those cases since, so far, the current approach is really just disease modifying at best and certainly not reversing. I want to shift gears on this concept of reversing and coming to neuron regeneration and neuron healing as a whole. And I guess part of me wants to start by, with what you've seen in your cell and animal models, do you feel like the approach of lowering dopamine in this case is sparking some sort of regenerative potential, or is it more so that it's stopping the damage and that there is an inherent healing capability of these neurons that's then taking effect, and that's why we're seeing a reversal of pathology rather than just a stopping of the progression?
I do believe that by reducing excess dopamine and creating the right environment, a shift towards normal physiology in the environment, both inside the cells and, maybe there's something outside the cells, but certainly inside the cells where dopamine excess is a problem, is a way to give these neurons a chance to reverse their dysfunction. So neurons are lost, they die. There's no controversy about that. People have done studies where, again, patients with Parkinson's who donate their brains or their families donate their brains and they're examined right after death, people count the cells in these regions. We know the cells are lost. We also know that the machinery, if you will, the energy supply for these cells is dysfunctional. That's the mitochondria. The mitochondria in this part of the brain, the striatum that's referred to, sort of as a quasi all-encompassing statement for that region of the brain, is an incredibly complicated network of neurons that some people claim has the highest density of mitochondria in the body, which means biologically there would be the highest amount of mitochondria in a region that has the highest amount of energy requirement. And we know that when you develop Parkinson's or in models of Parkinson's where there's excess dopamine, that the mitochondria don't function. They start to swell, they don't produce enough energy, they're still active, but their activity doesn't produce the energy. If we can reduce the toxin, based on some of these studies showing, like one of the studies showing the improvement in movement of the Drosophila, there's something that's allowing movement to come back towards normal. And I think it's that these neurons are smarter than we are. They know how to fix themselves better than we know how to fix themselves. Mm-hmm. Sure, we can do genetic sequences and we can, look at biochemistry of these cells, but we understand so little of how cells work. And sometimes what you need to do is be humble and say, hey, the cell's smarter than I am. Let me fix the environment and see if function restores. I may be biased by my experience from that beta blocker trial in heart failure, because that was part of an experience over about 10 or 15 years where we learned, because you can do heart biopsies in people, you can't do biopsies of the brain of this region with people. There's not enough cells there to undergo a biopsy. But we would see that the cells would function normally, the genetic programs would shift back towards normal. So, and cardiologists know that sometimes when you have bad coronary disease, part of your heart isn't moving, but it's alive. And you want a bypass surgery or a stent placement. And you can take what we refer to as hibernating heart muscle and get it to be functional again. And I think that by fixing the environment, removing the excess dopamine, and it's not dopamine itself that's the bad actor, it's the toxic breakdown products of dopamine that are definitely bad actors. We get rid of those or reduce them enough, the cells are going to be smart enough to figure out how to start functioning again, and people are going to be able to function better.
I wonder, this idea of hibernating, I wonder if there are a lot of neurological syndromes out there where the nerves aren't actually dead and gone, or that not all of them are, that we might think are, but rather they're hibernating or lower functioning, and that if we can create the right environment, they too can regenerate or heal, wake up, if you will. I'd love to move in the direction of getting your advice on generating cross-syndrome collaboration, getting people to work and discuss things outside of their silos of their particular indication, syndrome, trauma type, if you will. Because I know you had some experience with doing that within the FDA. If you were in my seat trying to get a larger focus on neuron healing as a whole, that which would potentially apply to across many different syndromes and trauma types, what would your advice be?
So I'll answer that question from 2 points of view, or two factors that color my view. One was my experience at the FDA, where I was brought on board in the Center for Devices to help transform the structure of the center to be a matrixed organization so that we would break down barriers. And so, you'd have a hierarchy in this direction with the head of X, and then the co-head of X, and then the people who did X, and the same thing over here for Y, and the same thing over here for A, B, and C, and create a way for people across all those silos to actually work together. And then I'll also talk about what I'm doing now, which is being a neurologist as someone trained in cardiology. So I'm trying to break down and bring some of my experiences in cardiology and also my experiences in regulatory oversight of drug and device development and bring them into the neurosciences of Parkinson's disease, taking into account what I was unfortunate enough to witness with Ivan as he continued to get worse. And, anybody who has been through this knows that there's a lot of dehumanizing that people experience. And for better or worse, he wasn't affected cognitively. So he experienced all that and not in a good way before he passed. So if I put all those things together, I think there are a couple of things. One is it's hard to find people who are willing to listen. And I think you need people who can listen and ask questions. I remember hearing a talk a few years back about improv and how the theories of improv work. Good people at improv don't start their turn by saying, but. They start their turn by saying, yes, and. That's their attitude. So you need people who can listen, who can say yes, and then what I've learned from this project in Parkinson's is you also need people who are comfortable looking like idiots, being wrong. I mean, I'm pretty good and I can have a conversation with just about any Parkinson's expert at this point. I won't necessarily convince them or win the argument, but I can engage really pretty well at this point, basically having self-taught myself Parkinson's disease physiology and pathophysiology since the end of 2017. So it's been a while, and for the last year and a half, it's really been my primary focus. So, being able to listen, having an attitude of saying yes and not being afraid to be wrong, I think are the kinds of people that are most likely to serve in a cross-silo environment and create a productive environment.
Fantastic. And then I'd like to finish with something that you mentioned earlier that caught my attention. You were talking about first principles, and it made me wonder, and this would be kind of a question maybe for the audience as well to just think about. Are there assumptions made about neuron regeneration and the healing capacity of the adult neuron that might not be accurate or are based on maybe unproven assumptions? What are your thoughts on that?
I think it would be hubris to think the answer was no. Of course, there are things we don't understand. I mean, that's the way I go into situations all the time. That's the way, I mean, when I was in cardiology, I was in the inner circle of where the knowledge was being created, working with unbelievably smart people. And one of the things I learned is you don't know, even if you think you do. And the importance of looking for data, the importance of playing devil's advocate against yourself and being able to objectively step back and say, here, let me prove I'm wrong, especially as you're dealing with those first principles. And really going back to that over and over again. Every time you take another step in your understanding about a disease, go back to first principles. I think that's the mistake that was made in Parkinson's. We kept getting smarter and smarter, but never realized that by the 80s and 90s, we knew things that undermined those initial assumptions, but nobody ever looked, or nobody wrote about it anyway. And nobody's come forward to me to tell me this is what they thought all along. So I do believe that there are first principles that are going to be proven wrong. There might be other approaches that end up leading to the same model, but I'm sure the initial assumptions are wrong in so many of the things that we're trying to fix.
Yeah. Great place to end, I think, with some good thoughts for those listening in, and to challenge the conventions, and which have in the neuron healing and regeneration world we've so far come up a little short, even though there have been some amazing breakthroughs in knowledge and progress in different areas. Certainly a long way to go. So thank you for those thoughts. And I feel like this is probably just the end of the first of many interviews. I'm really excited to stay in touch, and hopefully we can bring you back on as this phase 2 trial has some initial data, or even before then, if you feel like it would be helpful to give the audience an update, because this is obviously a really, really big part of what we're looking at when we look at all the different forms of neurodegenerative disease and neurotrauma. Parkinson's is a huge one. I lost my grandfather to Parkinson's, so it's near and dear to my heart as well. And I really wish you all the best moving forward.
That's great. Thanks. I look forward to continuing the conversation. Thanks so much for this.
Absolutely.